It depends. Okay, let's see. What am I doing? We need CD cards. Good morning, good morning. Wait, 12:06. Wow, I can't believe it's 12:06. Okay. And I also need an eye clicker. Go, and then things.
Ask a question real quick, or you want to wait till
Which guy?
it says I didn't do the question click.
Oh,
Yeah,
oh.
I just had like.
It's because it was the conclude. I was grading and grading. Okay, so don't walk out of the class,
Okay.
and I'll fix it. Okay, so just hang on to it. Okay. All right. So we were really starting section 2, right? Feel like I don't remember all this stuff. Lab reports are up here if you need if you have a lab report. I'm not logged into iClicker. I started it, but I did not log in.
It's okay.
Thank you for reminding me. Sometimes I start it and then I just don't finish it. Okay, so now you should be able to click in, check in on iClicker. So make sure you go ahead and do that. Okay, so we were on slide 11.2. Full screen. Yep. Okay. All right. So a few things. So if you're in my lab class, I have graded lab reports ready to go for you right here. But the grades aren't posted yet for lab. Grades are not posted yet, but they are graded, and I did write them down so you can take your lab report. For our class, I have all the exams that you took on Friday graded miraculously enough, and I did post those grades to Moodle, but I cannot give them back until Wednesday. So Wednesday, I will give you back your exams. Lab reports are right here. If you're in my section, yeah. So I will give back the exams on Wednesday, but your grades are posted, and there was a curve, but the average was really, really high on this one. So y'all did a really great job. So questions on that? Good. Okay. All right. So yes. For lecture or lab? For lecture, it is not weighed differently. It is 100 points, just like a regular exam, but it is longer than a regular exam, and it is cumulative. I just don't like, excuse me, making the final exam a huge portion of your grade. I don't like your grade being dependent so much on one exam. I just don't like that, so I don't do it to you. About double, maybe a little less. What about double? I know, but you got this. Look, I know it sounds like a lot. I know y'all, I just said double and people like about had a heart attack, but don't have a heart attack, because y'all are gonna realize when you are studying for the final exam that there are lots of concepts that we continuously revisit the entire semester long. So you're gonna look back on that stuff in the beginning and go, oh, I got that. right? So you're going to be surprised at how much you know. So do not freak out, okay? Study, but don't freak out. Sure.
One, if information was not covered on a test, is it
If it was in lecture, it could be on the final.
Okay.
Yes.
And then will you be like pulling questions from old tests, or it'll be completely
No,
new?
it'll
Or?
be different.
Okay.
It'll be different. Yes, it is. It'll be different, but it'll be the same concept.
Yes.
You know, I'm not going to ask you exactly the same question, but I am going to test you on the same thing, like glycolysis. I mean, you know,
Information was covered.
right. Right, but the more we really talked about it, the more likely it'll be on the exam. I mean, I'm not going to pull obscure stuff for the final exam. I'm not going to do that to you.
Like a study guide. Or something that you give.
Yes, yes, sir. No. No. Rob? Are you going to need a calculator? Yes. For the final exam, you do need to bring a calculator. Absolutely. Absolutely. Anybody else? Lab reports over here.
Speaking of the math, is there like, prior to the exam, is there any way we could go over what potential like types of math questions or something like that on the test?
On the lab final?
I'm sorry, lecture.
Oh. So the really good place to go to look for like what kinds of problems, a lot of times are the problems that we did in lecture. So if you go back to the lecture notes and you look at the ones that we actually solved the problems in lecture, that's a really good one for what's going to be on the exam. And then if you look in the book, sometimes they have some duplicate problems similar. You know, like if you want to try, okay, we did, I don't know, membrane potential or whatever calculation we did, usually they have extra in the book. So if you look at the end of the book, the end of chapter problems, they're going to have some in there too. And usually, not always, but usually there was a similar math problem in your homework too. So if you've seen it in multiple places, like as you're reading the book, in lecture and homework, it's probably going to be on the exam. You know. Okay, did that answer your question? Okay. Anybody else? Good. Ready to go? Okay, so we're really going to get into the nitty-gritty of the electron transport system. Now, before we get into the nitty-gritty, I kind of want to talk about it and kind of draw it out just a little bit with you guys, because it's a lot of information all at once. So there aren't a whole lot of slides for this section, but it's a whole lot of information, right? So in the second section, we're really going to look at 4 functional things. So we're going to look at the 4 functional components of the electron transport chain, right? So if we break down the electron transport chain, I know that we can break it down into complexes, but that's not functional. That is not a functional definition. So if we look at which ones are large multi-subunits, so large multi-subunit complexes, right? So this is complexes 1, 3, and 5. Now, I don't want to use those terms all the time. I don't just want to say complex 1, complex 3, complex 4. I want to actually say their names. And you're going to see when we name them that their names will actually make sense, which is awesome. So where do we drop electrons off from when we start at complex 1? Who brings electrons to complex 1? NADH. So we're going to name it NADH. That's the beginning of the name. Ho, ho, ho. Pretty nice, right? Okay, now, where are we going to give electrons to from complex 1? We're going to give them to Q, or another name for Q is ubiquinol. Q-U-I-N-O-L. N-O-N-E, ubiquinone. And so what kind of a reaction is going to happen? It's going to be an oxidation reduction reactions, right? So we call them oxio, O-X-I-O, reductases. So this is NADH ubiquinone oxioreductase. Hey, that kind of makes sense, right? Not too bad. Okay, now the next complex, the next big complex is complex 3. Complex 3, right now we have our electrons in ubiquinone. So this is ubiquinone. And then the next place we're going to send those electrons from ubiquinone, and ubiquinone is another name for Q. Somehow you're going to see them interchangeably in the text. So you just have to kind of know that. As we're going to give those electrons to ubiquinone, and then ubiquinone is going to pass them to cytochrome, cytochrome C. So the name is ubiquinone, cytochrome C, and then what kind of a reaction is it going to be? Oxidation reduction, so what should we call it? Oxioreductase. Isn't this nice? Like they look like really complex names, but the names are telling you what's happening. Okay, then the last one, we're at cytochrome C. And this is our final destination. This is where we're going to convert oxygen into water. So all we do is we say this is cytochrome C oxidase. That's the end of the road. We don't need to keep passing things along. Not too bad, right? Hey, those are pretty nice. Okay, so those are our big 3. And the kind of the thing to remember about these big 3, these are going to traverse the inner mitochondrial membrane. Means they're embedded in the membrane, but they actually still have to have functional components that are going to be exposed to the matrix, but they really traverse that membrane. They go across the whole membrane. And what do they have to do? If this is part of the electron transport chain, in order to drive ATP synthesis, what are we trying to put into that intermembrane space? Protons. So they have to be proton pumps. There's actually PUMPS. There are actually more than one way to translocate protons. A pump is one of them. So if you just want to say are proton translocators, that's probably a better word to use. And we'll talk about that today, why it's not necessarily a pump, but it does translocate things. Okay, so that's one functional component. Yes, ma'am.
Why are they called oxidases? They are.
Well, so you have to think about you're not just doing an oxidation reaction. In them, you are doing oxidation and you are doing reduction.
So why is it called oxidase when everything else is called oxioreductase?
Because you're at the end of the line. We're talking about our final destination of our electrons, right? What are they going to be used for finally? Convert.
So it's just used for like signaling that's the end.
Yeah.
Okay.
Yeah. Mm-hmm. Okay. So we got one functional component, but that's only one functional component. Oh, goodness, right? We got 3 more to go. So the next functional component, right, is those mobile electron carriers, right? We said we're going to convert it from one complex to another complex, right? Well, the complexes are stuck. They're not going anywhere, but you have to have something mobile that can bring things over, that can move within the membrane. So the next one is coenzyme Q, which we call ubiquinol. Ubiquinol. So this is small, hydrophobic electron carrier, right? And so it's going to move laterally from one complex to another, right? So diffuses laterally. What do I mean laterally? Like left to right within the membrane. It always is going to stay in the membrane. And it's going to donate electrons from where? Well, where did the electrons start? If I'm in ADH, where am I dropping off electrons? What starts the process? Complex 1. But that's not the only possibility. I could be, instead of NADH, I could be what? FADH too, right? So then where would I be dropping off electrons? Okay, so I have electrons at either complex 1 or complex 2. So either way, Q, right, will donate electrons that came from 1 and 2 and transport to 3. Right, so you can dump off electrons at 1 and 2, but Q is going to take them to 3. Yeah. And it actually carries electron pairs. That's a very important designation. Okay, didn't I miss a complex? Did I miss a complex? Well, no, so I did 1, 3, and 4. So I missed complex 2. So you have complex 2 is an example of a membrane associated enzyme. If I could write today. Membrane associated enzyme. So one of the membrane associated enzymes is complex 2. Complex 2 is actually succinate dehydrogenase. But there are actually multiple membrane associated enzymes. So do you remember when I told you we actually had to shuttle in the electron carriers from glycolysis? Do you remember that? And I said if it was in the muscle, you would get a little bit less ATP per molecule than if you were in, for example, the liver, right? Remember that? Okay, so the shuttle system is one of those membrane associated enzymes. So we'll say the glycerol 3-phosphate dehydrogenase, dehydrogenase shuttle system. Okay. And then the very last one, do you remember how we said, okay, if you go on a low carb diet, where does your body get the acetyl-CoA required for citrate cycle and then therefore electron transport chain? Where does all that energy come from? Fatty acid metabolism. So we need to have a place where those electrons from fatty acid metabolism can actually enter the electron transport chain. And so it's called the ETFQ oxioreductase. Oxioreductase enzyme. So this is from fatty acids, fatty acid only. I don't know why I put an extra O. Right. So here, right, you're going from fatty acids to acetyl-CoA, right? And you get those 2 electrons from that transfer. So what do all of them have in common? What's the job of all of these? Yes, they shuttle what? Well, but what is FADH2 carrying? Electrons. So all three pick up electrons and donate them to Q. Okay, oh my gosh, we have one more. We're almost done. We're almost done. This is the last functional component of the electron transport chain. We didn't talk about one of the electron carriers. What's the one electron carrier we hadn't talked about yet? Cytochrome C. Cytochrome C is all in its own little category for a couple of reasons. It's on the cytosolic, so solic side of the membrane. And it does carry electrons like Q, but it only carries electrons one at a time. And that's kind of a problem because everything else carries it two. And so we'll have to do some very special things to make sure that it can carry them one at a time. And it's going to carry electrons one at a time from where? Where do we kind of end up with our, we had Q goes to where? Cytosolic side of the membrane, right? There's the matrix side, there's the cytosolic side. Complex 3 to complex 4. Okay. So before we go into like some really, really crazy diagrams about how all this works, I kind of want to take what we wrote here and turn it into a little diagram. Okay. And then I think when you look at the slides, it's going to make a lot more sense. Yes.
Explain, I guess, what exactly Q is. Instead of this Ubiquinol,
Yeah.
What exactly is it? Is it
It's an electron carrier. It's an electron carrier, just like cytochrome C, except Q carries electrons 2 at a time. Cytochrome C carries them 1 at a time. Yeah. Okay, so we're going to start at complex 1, right? This is one of those big complexes that are multiple subunits and all that kind of stuff, right? Where are we getting electrons from? NADH. We're converting to NAD plus, right? It's actually plus H plus. But so when this happens, right, we're going to have a couple of different things happen. We're going to move some electrons, right? So I guess we'll do electrons in red, right? So we'll have electrons and they are going to get moved into something else. So where are they going to go? If this is embedded in the membrane, right? This is embedded in the membrane. We know that we have complex, I want to erase this. I know that I have another complex here. Do these electrons go to complex 2? They skip. They actually skip. So we'll kind of draw them going behind. They're going to skip complex 2. And they're actually going to go where? They're not going to go to complex 3. They're going to go to Q. Right? So you actually have a Q molecule really close to complex 1, but we're just trying to show they're all going to Q, right? So these skip 2. I can draw that. They skip 2. They go straight to Q. Okay. Now what about complex 2? Where do things come in at complex 2? right? And we're going to pick up electrons there. Where are those electrons going to go? They're also going to go to Q. You see how Q is kind of our central place where everything's going? Now, there are a couple of other ways that you can get to Q. We said 2 other ways up above. What were the 2 other ways that we said you can get to Q? If you undergo fatty acid oxidation, right? So we'll just put fatty acid oxidation. You can go through what? ETF, the ETFQ complex, right? So this is when we have those fatty acids and they get oxidized and they give up those electrons and they go to acetyl-CoA, right? So they're going to drop off electrons, and where are those electrons going to go? To Q. You notice the theme? Okay, then we also have our shuttle. So our G3P shuttle, right? That's also going to give us electrons that are going to go to Q. Is Q important? Absolutely, right? I think people think about the electron transport chain as a linear thing, and it's not really. Okay. All right. Now, what happens to Q once Q picks up those electrons? It's also going to pick up hydrogen atoms, and it turns into QH2. So this is with electrons, QH2 right there. QH2 has 2 electrons. But what did we say about cytochrome C? You can only carry them 1 at a time, right? So when we go to complex 3, the whole point of complex 3 is to split those electrons up, right? So here are our electrons. So we need to send 1 electron at a time to cytochrome C. Cytochrome C. So this will be, we'll put a little note, 1 electron, right? And so we'll split them up, and then they'll go 1 electron at a time to cytochrome C. And then where is it going to go? So our very last place. Yeah. I can make room. We're going to have complex 4. Right? And so those electrons are going to be sent to complex 4. Now, what's the whole point of having those electrons? What do I want to happen? O2 converted to water. Now, I'm just following the track of electrons. There's something else that's happening that we have not drawn here at all. What is the point of the electron transport chain? To eventually drive ATP synthesis. How do we do that? By making a gradient of protons. That's what we talked about all the last class period, right? Not the last one, because we had an exam. Time before that, right? So some of these complexes are able to pump protons. So if we look, we're going to pump 4 protons via complex 1. We're going to pump 4 protons, complex 3, right? And then in complex 4, we're going to be able to pump 2 protons. So if you're NADH and you come in, how many protons are you going to be able to pump across the membrane? You're going to start here at 1 and you'll be able to pump 4, 4, and 2. So how many? 10. If I am FADH2 and I enter here at complex 2, or on the shuttle, or on fatty acid oxidation, how many can I pump? Only 6. So now do you see why FADH gives you a different number of ATP molecules than NADH, right? Because it's all about where they're entering the electron transport chain and how many hydrogen ions they can pump. Cool? Now you have a good picture.
So you said that for QH2, that Q transported 2 of them.
Yep.
When it, is it Q still is able to transport 2, but when it just gets to.
When it gets to 3, three's point is to split this into being 2 electrons at a time to being 1 electron at a time. That's the whole point of complex 3. And in that split, you'll pump hydrogen ions. Yep. Okay. All right. Now y'all ready to get into the nitty gritty? Do it. You got this. You got this. Okay. So this is just a very, very much a prettier picture than my drawing. But when you draw it yourself, each step, it's going to make a little bit more sense. Okay. So if you can try practice drawing these diagrams, it's going to help you to understand it. Right. So here's complex 1. We said NADH enters complex 1. So we have NADH plus H plus. The NADH being oxidized or reduced. We lose hydrogens. We lose hydrogens, oxidation or reduction. So oxidation is a gain of oxygen or a loss of hydrogen. All right. Okay. So now those electrons, so here are 2 electrons. Those electrons get passed along. They're just showing this as a single line, but we're going to talk about it in detail. But they get passed along by a bunch of different complexes, a bunch of different things inside of complex 1, and eventually get picked up by Q. To convert Q to QH2, I need to, number 1, pick up 2 electrons. And what else do I need to get to QH2? 2 protons. So 2 protons are picked up here. So in this whole process, we're going to pump 4 hydrogen ions from the matrix to the intermembrane space. Okay. So now I've pumped my 4 hydrogen ions. I've taken my 2 electrons. I've stuck them on Q. I've given Q 2 hydrogen atoms. I now have QH2. QH2, 2 electrons at a time, get sent to complex. What complex is that again? Is that complex 2? No, that's complex 3. Good. So QH2 will bring those 2 electrons to complex 2. Now, do you see how when they came in, they were coming in 2 at a time? Now they're going to leave 1 at a time. So cytochrome C is actually going to have to make 2 laps in order to take care of those 2 electrons. And in that process, we have 2 hydrogen atoms that are pumped, protons that are pumped out. But why is it 4 here? Where did the other 2 come from? Q. They came from Q, from QH2. So you have 2 here already, and you have 2 here for a total of 4. Cytochrome C takes those electrons to complex 4. Complex 4, these electrons are used to convert oxygen to water, and we can transport the last 2 electrons. So that's what creates our proton gradient. Now, here are the other ways that you can enter the electron transport chain, because that's not the only way, right? What if you are something else? What if you are NADH, right? When we convert from succinate to fumarate in the citrate cycle, what do we make? FADH2. So our FADH2 is carrying 2 electrons, drops them off in complex 2. Complex 2 can't pump anything, so that doesn't give any energy to the system. But it gives them to Q, and then we can go on with the rest of the pathway. You could also get them from the ETFQ complex, which will take our fatty acids and reduce them to acetyl-CoA. So we can get 2 electrons that way to send to Q. Or we can get 2 electrons from our shuttle system, right? Doesn't matter. They all go to Q, and then they pick up with the rest of the pathway. So this is just showing that there are 3 different other ways to enter the electron transport chain. Okay, but we kind of already knew that. All right, so you got to think about if something happens to this whole system, what's the net effect? If you stop the electron transport chain, what's the net effect? Yeah, you stop the proton pump. You stop driving ATP synthesis, what happens to your cells? Cell death. This is cell death. So when you look at some of the various inhibitors that will inhibit the electron transport chain, rotenone is a compound that is used on poison tip darts in some cultures, which can be very, very lethal. Hydrogen cyanide. Hydrogen cyanide is one that was used actually in the Nazi gas chambers. So in this one, you're actually blocking complex 4. So they would drop sodium cyanide into sulfuric acid, right? And it would produce the gas, the hydrogen cyanide gas. And that binds to things inside of the heme groups inside of complex 4 and basically shuts down complex 4. So very quickly, you stop driving your proton pump, you stop ATP synthesis, and you die. So it can be really bad. We call carbon monoxide an electron transport system inhibitor. Why? What do you need at the very end of the electron transport system? Oxygen. And so CO, carbon monoxide, binds to hemoglobin, we know this, with like a 250 times affinity than O2. So if carbon monoxide is around, your cells are going to bind carbon monoxide preferentially over O2. But if you no longer have a final electron acceptor, you shut the pathway down, right? It's a cyclic pathway. You have to keep going. You're going to shut it down there, right? Antomyosin A is a chemical that is made by a staph bacteria, and that staph bacteria, we were able to isolate it, and it's actually used as a toxin in order to get rid of non-indigenous fish populations. It's really kind of interesting. But there are lots of different applications for inhibiting the entire system. But if you want to understand how the system works, what is the only reason why electrons make all these jumps that we've talked about? Why are they moving from one thing to another? They're standard reduction potentials of all the different compounds that are involved in the process. So take a look. We're going to start, let's say, with NADH, and we want to go all the way to the end to our final electron acceptor of O2. So what happens to that standard reduction potential as we go from the beginning of the pathway to the end of the pathway? It slowly goes up. Now, is it a perfect increase? Not quite, but these are standard reduction potentials. So are they perfect to what is actually in the cell? No. And the concentration of the individual components will have an effect on their real reduction potentials. So you have to kind of keep that in mind. But if you look in general, we go from something negative to something positive, right? So you kind of keep that in mind. Okay, so how do all of these oxidation reduction reactions result in moving hydrogen ions to the other side of the membrane, right? Well, there are technically two different ways that we think this happens, and I'm going to repeat that. These are the ways that we think it happens. We've got some data to support it, but like we don't quite understand it, which is kind of interesting. I always like pointing that out because, you know, you always sit in these classes and you think we know everything, right? We don't. We really don't. But this is kind of how we think it's happening. So you can have something called a redox loop. In a redox loop, you start with a compound that has hydrogen atoms and it has electrons. So that's this XH2, right, that you see. When it enters this complex, right, what happens is you're going to actually separate the protons and the electrons. So the protons from this original substance, the H2, are going to get stuck onto something else. In this case, it's Y, right? What is it for real? What do we think this really happens in? Q. We think this really happens in Q, right? So Q is going to pick them up and shuttle them over to another complex. So what's going to happen is that those electrons go along with Q, the hydrogen atoms go along with Q, and when we get to complex 3, those hydrogen ions are split off. The hydrogen ions go on one side of the membrane, the electrons go on the other side of the membrane. So the electrons are used to reduce something in the matrix. What gets reduced in the matrix in complex 3? Well, Z in this picture, but what is it for real if we're talking about complex 3? Right? If this were complex 3, and this were really Q, right? So your electrons are being picked up, could be by cytochrome C, but eventually, where are those electrons going? They get passed down to complex 4, and what do they use to change in the matrix? What changes in the matrix? 3 to 4, and then we go O2 to H2O, right? Okay, all right. So the other way that this could happen is through what's called a proton pump. This is kind of more of what you guys are used to, right? The initial step is exactly the same. We have some substance that's carrying electrons and it's carrying protons, right? So we send it off to Q, Q picks it up. But here in this case, what's actually happening is that those protons are transferred along with the electrons. But this activity right here, induces a conformational change in the complex. And that conformational change in the complex changes the pKa values, values of the amino acids, the functional groups of the amino acids, right? So if you start changing pKa's of amino acids, what's happening? Not necessarily, but it's inducing a conformational change. In this case, it's a functional conformational change. So before we couldn't pump anything, we induced the conformational change, and now we can pump hydrogen ions. So it creates a functional change. A denaturation would be a non-functional change. Okay, does that kind of make sense? There are 2 different ways that happens. And we'll talk in more detail about these 2 processes. Okay, so let's kind of start at the beginning again. So what do we call complex 1 from now on? NADH, ubiquinone, oxidoreductase. Okay, so the NADH is what's oxidized and the Q is reduced. This is by far the largest complex. And when we talk about the largest complex, we're talking about 15 to 40 different polypeptides. Mammalian has 40. 40, y'all. That's huge. So all of these different complexes, what you're going to see is they have tons of functional groups inside of them. And I'm not talking about amino acid functional groups, more like prosthetic groups. Prosthetic groups are groups that are not part of the amino acid. So a vitamin, a lipid, a metal ion, something like that that can pick up those electrons. So one of those is Flavin Mononucleotide. Flavin Mononucleotide is really, really similar to FAD, right? And it has the same sort of function. It accepts electrons, but in this case, it accepts electrons from NADH. So if we look at how this works inside of our complex, right? We have NADH plus an H plus. The NADH converts to NAD plus, which means that we're losing our electrons, right? So those electrons are picked up first by Flavin Mononucleotide. Then these are iron sulfur clusters that then pick up those 2 electrons and slide them along, right? And why are they moving from one group to another? Why does this happen? The standard reduction potential as we go this way increases or decreases? It increases, it becomes more positive. Okay, so now we have these two electrons that in combination with two protons will convert Q to QH2, right? So that caused a conformational change in our protein, which now allowed it to turn into a pump to pump 4 hydrogen ions into the intermembrane space. So from this step, we've gotten 4 hydrogen ions into the matrix, and we've converted Q to QH2. Now, do we go on to complex 2 in order? No, where are we going? Complex 3. So kind of keep that in mind. They're not, that's why I don't like naming them complex 1 and complex 2, right? Okay, so FMN is one of those compounds that reduced 1 electron at a time. So if you look at this nitrogen right here and this one right here, those are the ones that are going to pick up the electrons and the hydrogen atoms. And so they pick them up just one at a time. And so they call this where you've only picked up one, the semiquinone intermediate. And then fully reduced has picked up 2 electrons and 2 protons. But you don't have to memorize structure. You don't have to memorize structure. Okay. All right. So now we've gotten our electrons to coenzyme Q, right? So coenzyme Q is a mobile electron carrier. It's not stuck. Complex 1 is stuck in the membrane. It can't go anywhere. But Q is mobile and will transport those electrons from complex 1 to complex 3. So it's also the entry point for electrons, we said, from citrate cycle, from fatty acid oxidation, and we said from glycerol 3-phosphate dehydrogenase, which is the shuttle system for glycolysis, right? Okay, so now we're going to convert, once we're in complex 3, we're going to convert our 2 electron transport system into a 1 electron system at a time movement, because that's what cytochrome C can do. Cytochrome C is super highly conserved across so many different species. So it's a very old protein, I would say. And so the system that we use to describe how those electrons go from being 2 at a time to being 1 at a time is called the Q cycle, right? The Q cycle. So what actually happens, right, when we look at Q is a similar sort of deal as the FMN, right? We have an oxidized form. We can pick up 1 electron, 1 proton. We get our semiquinone intermediate. Then we can go and pick up 1 more electron, 1 more proton, and we get the fully reduced ubiquinol. So you have to know that oxidized is called ubiquinone, reduced is called ubiquinol. So there's kind of like 3 different terms for this, right? So you have to know them. You do have to know them, okay? So let's look kind of at how that works. So the other way that we can get to Q, right, is we can go through complex 2, and this happens all the time from the citrate cycle. When we convert succinate to fumarate, we make FADH2. The FADH2 donates its electrons. This is another iron sulfur center, just like what we saw before in complex 1. But here, this is a cytochrome. This is a, I'm sorry, not a cytochrome. This is a heme center, a type B heme. And that heme will pick up the electrons and then send those electrons to Q, right? So this is the same enzyme that participated in the citrate cycle. It's not an additional enzyme. We're just calling it 2 different things, right? What did we call it in the citrate cycle? Succinate dehydrogenase, right? What do we call it in the electron transport chain? Complex 2, right? Which is why it kind of has 2 different names, right? It's not really a complex, it's really an enzyme, right? So be careful about that. That can kind of throw people for a loop. Okay, so complex 3, right? We're finally to complex 3. What do we call complex 3? Ubiquinol, right? Because it's the Q trying to send electrons to cytochrome C. So we call it the ubiquinol cytochrome C oxioreductase. So this is going to reduce our cytochrome C, but at the same time translocate 4 hydrogen ions to where? To what? Where are we sending hydrogen ions? What's the whole point? Enter mitochondrial space. Enter mitospace, right? So you have to have a docking site for Q, and you have to have a docking site for cytochrome C, right? Both of those carrier proteins are going to have to interact with complex 3. So technically, it actually contains 2 binding sites for ubiquinol. One of them is closer to the inner mitochondrial space. One of them is closer to the matrix. So this, remember the N, what does the N stand for? Negative. So this is really close to the matrix. QP is close to the inner mitochondrial space. And we'll see that in just a second. And so those electrons go through lots of different electron carriers. And in this case, it's iron sulfur clusters, just like what we saw before in complex 1 and complex 2, right? It's the same sort of deals. So how does this work? You remember when we loaded Q, we gave it 2 hydrogen atoms and we gave it 2 electrons, right? So we're at QH2. QH2 is going to come in and dock first here at the QP site. So it's close to the intermembrane space. It's going to lose first, I know it says 4 here, but it's going to go through 2 cycles. So you have to kind of stick with me. So we're going to have 1 QH2 come in, bind. It's going to drop off 2 hydrogen protons to the intermembrane space, right? But the whole point of this is to do what? The whole point of complex 3 is to do what? Go from carrying 2 electrons at a time to carrying 1 at a time, right? Because Q is carrying 2 electrons, so carries 2 electrons at a time. Cytochrome C carries what? 1 electron at a time. Okay, so we let go of our 2 hydrogen ions. Now we have 2 electrons. One of those electrons goes through an iron sulfur complex through to cytochrome C. That other electron is actually going to get passed around inside of complex 3 into those heme groups, right? And it's actually going to get picked back up by a Q molecule, right? So this is really temporarily Q, but it only has 1 electron, right? Is that QH2? How many electrons does it need? It actually needs 2 to be QH2. So I've basically given it 1 electron so far and 1 proton. Is it ready? No. So this Q, though, has dropped off 2 protons and dropped off 2 electrons. So where can this Q go? It's now free to travel back to complex 1. So I have a second Q that's going to come into the QP site. It's going to release 2 more protons for a total of 4. It's going to release 2 more electrons. One of those electrons goes to cytochrome C. The other electron gets transported back to this Q. Now I'm going to give it 2 electrons, 2 protons, and what do I get? QH2. Now this QH2 can be recycled and go back and bind on the QP site. It happens twice. Do you see how it has a 2 here? It happens twice. Yes, sir.
So what's the difference between really how it goes down in the complex?
Here.
Yeah, down. Does that and it goes back. So yeah, I get it.
So this is the first Q. This is the second Q. So watch, I'm going to show you. Let's see if it makes more sense if we break it down into 2 different ones. Let me see if I can zoom in. Come on. View, zoom. Oh yeah, 300. Okay, that's the best. Okay, right. So this is our first QH molecule, right? Now we're breaking this down into 2 diagrams of exactly the same process. We have QH2. QH2 comes in, binds to the QP site. That creates a release of 2 hydrogen ions into the intermembrane space, right? Now we have 2 electrons. One of them goes to cytochrome C. One of them gets moved along and gets picked up by Q, right? There was a Q. You got to think that this cycle is happening all the time. So there's a Q waiting, right? There's a Q waiting. And that Q that's waiting is going to pick up that 1 electron and is going to pick up 1 hydrogen, right? But then we have to have a second QH2 molecule. So what happens to the second QH2 molecule? The second QH2 molecule comes in, binds to the QP site, releases another 2 protons to the intermembrane space. Those 2 electrons get pulled off. One goes to cytochrome C. The other electron now goes to QH to turn it into QH2, right? But now I have a Q that's free. That one gets released and can go back to complex, whatever. It's not always complex 1, but it can go do its job again. So from this, I'm going to make a Q and I'm going to make a QH2. And then I'm also going to load 2 cytochrome C complexes. Yeah. All right. One more class. My God. Oh, I forgot. Before y'all walk out the door, I need y'all to complete evaluations online through Moodle. If 70% of the class does it, now I can't see what you write about me. I just want you to be honest. If 70% of the class has a response, I will give 5 bonus points to everyone in the class on their final exam. Okay? All you have to do is complete it. Please be honest and please be helpful. Just complete it. All I can see is percentage completed. So 5 bonus points on that final exam. Anybody on Zoom need anything? Oops. Okay, so just as good. Anybody else still on Zoom have any questions? Because we still got a few people on. Okay, so they must all be good. All right, y'all have a great day.
So that's what I focused on yesterday. So